Okay, now we move to the second speaker of the session, Professor Thomas Bosch from University of Kiel in Germany, with the talk Importance of Preserving Microbial Biodiversity and Evolutionary Informed Perspective. So thank you also, Professor Thomas, for accepting our invitation. And the virtual stage is yours. Thanks again.
Thank you so much. Can you hear me?
Yes, perfectly. Yes.
Perfect. Thank you so much. Thank you so much for the invitation. It's a great honor. And I lead you in the next 15 minutes a bit in a different direction. First of all, welcome from Germany. And second, I'm an evolutionary developmental biologist, and I want to bring your attention a bit to this more evolutionary oriented aspects of microbial diversity.
Thomas, we see your not the whole. We can see the speaker's view. Probably you can make it the whole slide view.
Okay. On my computer is the whole thing.
We can see the two. It's okay, but it's smaller.
It's the one at the top. I don't know in German. One of those three buttons at the top.
What should I do?
Right.
You have to duplicate the displays.
Pardon?
Duplicate the displays up there. One of those buttons should change the view, but I don't know. It's
okay.
Which button?
Yeah, yes. Now, perfect. Thank you so much.
Okay. Thanks for the it's very clear to all of you. Microbes are there and were there on Earth a long time before any other animal, plant organisms existed. It was astonishing. A new knowledge to me many years ago when I learned, because I'm working on presumably very ancient organisms, that 3.5 billion years the Earth was colonized on all kinds of substrata, marine, terrestrial, in the air, by microbes. And so the microbiology is really woven into the fabric of complex life, and even the origin of the eukaryotic cell, the mitochondria, the chloroplast, etc. All of that is just microbial achievements. And so nothing really what we see today on our Earth can be understood without having the microbes taken into consideration. And with that, it's we thought about this fact a lot and decided that actually organisms, and I'm sure you will all agree, organisms cannot be considered as an individual organism without at the same time also considering the microbial parts, the viruses, the bacteria, the archaea, the fungi, symbionts, which are part of this organism and which actually determine the fitness of this organism. And so we call this synergistic, interdependent gemisch of different organisms a meta-organism or a holobiont. And that's actually, I'm directing a center here in the Kiel University on origin and function of meta-organisms. And I think for us all, it's very clear that's the animal condition to have microbes. There are no germ-free animals and germ-free plants around in the world. It's also clear, and it was already said at the beginning of this meeting, that biodiversity is critical to life on Earth, and that we of course face at the moment a major crisis of loss of diversity. About 1 million species of plants and animals are threatened to extinction. It's one of the major challenges which we have in our time and which we somehow have to cope with if we want to continue to live on this planet for a long time. It's also very clear that, and it's a lot in the press and in news and in scientific articles, that the reasons for the decline of this biodiversity, there are several. This is habitat loss because of climate change, invasive species, pollution, overexploitation, and others. The big question mark, or what we miss and what I miss, is that normally, although there is a lot of news and talks about biodiversity, we have to face, in particular, you and me, as microbiologists and microbial diversity experts, that in the general news, microbes are really not yet existing. And when we talk about biodiversity loss, and this is just one of these cartoons here. You can look in many different aspects. If you give it the keyword biodiversity, this always refers to the decline in the number of plants, of animals, of insects. But it's very clear that macrobiodiversity and the loss of macrobiodiversity is based on microbial diversity and the loss of microbial diversity. And when attending national, international conferences on the topic of biodiversity in Germany and elsewhere, I sometimes have to sit for 3 days in these meetings, and I don't hear a single time the word microbes. I feel a bit strange, and I feel a bit that we have to do something to change that, because the microbes and the loss of the microbial diversity is, of course, the issue which counts here. And what I would to do with the second part, and so this very short talk, is I would just give you one recent example. What does it mean, loss of microbial diversity in an organism which is among the oldest multicellular organisms on Earth, and it is believed about 560 million years ago. It's a cnidarian, it's a basal metazoan, it's a freshwater polyp hydra where I work all my life on. And we always thought that this organism is, represents the and ancestral situation of an organism, and that's why it's interesting. We discovered some time ago that this organism is colonized by a very stable microbiome. It sits in a mucosa which is made by the same mucins as in your mucosa in the intestine. It's a rich diversity. We can culture all of the bacteria. We can genetically manipulate them. We can genetically manipulate the host organism. So we have a relatively good understanding of the microbiota in this organism. And what I would like to ask now for the remaining time which I have here is with one example, and I could give you many, and we have published that. That just the example which I show you just came out a few weeks ago in Current Biology. What happens if this microbial diversity is reduced? And to answer the question, first of all, you need gifted people to do the work, and the work I'm presenting in the next few slides is the work by a gifted graduate student, Christoph Gietz. And we found, to our big surprise, that a basic feature of animal behavior and human behavior, of course, eating, is heavily dependent on a highly diverse microbiome. And if you reduce the diversity, the eating reflex, which these animals have, is heavily disturbed to the fact that they cannot open the mouth anymore. And I give you the examples. I show you that. This is a cartoon where it shows that when these animals are eating, so they take some shrimps, and then the mouth opens, and then the shrimp goes into the gastric cavity, and then it's digested. And what we are studying, and what we have studied in this paper, is the mouth opening in the presence or absence of the microbes. And we do that. We do that by calcium imaging and transgenic animals. So what you have here is you have in a certain subpopulation of neurons, you have a calcium indicator, and each time the mouth is opening, then these neurons start, as right now here, they start to flash, and we can induce that by a chemical, glutathione, so we don't have to feed the animals anymore. And now you can beautifully study what happens if the animal has a microbiome outside, or a microbiota outside, or it doesn't have this microbiota outside. And first of all, Christoph Gietz uncovered and characterized the neuronal control system, which is involved, and with all the neuronal subpopulations, so there are 7, and there comes one subpopulation which gets a signal, and then the signal is transported to another subpopulation, and at the very end, the mouse is opening, the whole thing takes about 1 minute. And the big question, of course, is what happens if you have germ-free animals and when we have animals which have a disturbed microbiome. And as you can already see in this cartoon, there is a dramatic influence of the microbiome. And the influence is when we remove all the bacteria in germ-free animals, the mouse opening time is drastically reduced. But if we disturb the microbiome, that we do not remove all the microbes, but leave one colonizer in the microbiome, which is has the name Corpibacter, then the animals cannot open the mouse anymore. So they are colonized by a bacterium, but the bacterium has not the other members of the microbiome, and you can then the mouse cannot open, and you can rescue that by giving back the high diversity microbiome, the rest of the bacteria, one by one back. So then the effect is gone. And so that's all what I wanted to show you. The conclusion is, of course, I have to give you the conclusion that a diverse microbiome has no effect, but reducing the diversity to one member has a strong inhibitory effect. And in this paper, we have shown that this bacterium produces glutamate. And if there is only this bacterium there, because we have removed the rest of the diverse microbiome, then this glutamate is accumulating and it binds to a neuroreceptor, and this neuroreceptor is activating muscle epithelial cells, and this is then causing the disturbed mouse opening. Normally, if there is a high diverse microbiome, this glutamate is eaten and taken away by other microbes because they need it for their metabolism. So in a normal microbiome, of course, the animals can beautifully open their mouths. But if you reduce diversity, they cannot open the mouth anymore. Just want to remind you that glutamate is present in nearly every food, what we take and what we buy from the supermarket as an appetizer. So coming to the end and to some thinking at the very end, at the moment, the knowledge of loss of biodiversity is mostly limited to macroorganismic studies. However, the fundamental elements that lead to the loss of biodiversity are found in the microbes. In my personal academic life, there is a book which came out in 1992 by Peter Lawrence, The Making of a Fly. And this book proposed at that time that we have understood how to make a fly. And that was driven by the fact that we have understand the fly genes and the homeobox genes and the bow plan and everything. But just, and this is added by me now, just what is completely ignored in developmental biology still today is that beside these 13,000 fly genes, the fly has 120,000 microbial genes. And how can we ignore 90% of the fly genes, which are microbial fly genes, in development, physiology, and evolution? We have to change that, and we have to educate our colleagues in developmental and evolutionary biology that only by, as somebody said already, by a holistic understanding of the meta-organism, we really get to a complete understanding. That is the task, and I admire Gloria's and Marty's and others, the Microbiome World Initiative. That's why it is so essential to preserve the microbial diversity, to characterize the diversity, and also to study it functionally. With that, I thank my collaborators. I have a wonderful team in the lab. This is the center of the Collaborative Research Center funded by the German Science Foundation, where I'm sitting right now. I thank the German Science Foundation, and I thank you very much for your attention.
Thank you so much, Professor Bosch, for the amazing talk and also for staying to the time. Well, I think it's really, really nice to show that how holobionts could be our opportunity to convince the rest of the scientific community that the microdiversity is important to be studied in many different organisms, but also in the environment, which is always a little bit more complicated to convince to the rest of the world. So thank you so much for that talk and this informed perspective that you are giving us.